Thank you very much, Chair. First of all, my sincere thanks to the organizers of this workshop, especially Rajiv, for this wonderful opportunity to be part of this very exciting program. What I'm going to talk is the collective effort of several colleagues in the CIMMYT Global Maize program, as well as an array of institutions which are partners with CIMMYT in Sub-Saharan Africa, Asia, as well as Latin America. This is something which is not just specific for maize, but one that affects millions of smallholders across the tropics. The challenges are indeed real with regard to climate change, especially in terms of drought, flooding, and heat. Biotic stresses, particularly the pathogens and insect pests that are affecting the maize productivity in the tropics due to several reasons, including the new cropping systems that are emerging, which is posing additional pressure. The declining water and nutrient resources in the soil is again something which we have to deal with extensively in the tropical regions, and coupled with a variety of factors that are leading to soil degradation, erosion, and nutrient deficiencies. CIMMYT's strength has been this nearly 4 decades of breeding for abiotic and biotic stress resilience in the tropical and subtropical regions. And this effort started way back in 1975 when managed stress screening approach has been introduced, and several cycles of recurrent selection have been exercised on an array of populations, especially what you see here quite often, Laposta secchia, Tuxpanno secchia, Pool 16, Pool 18, and so on and so forth. And this has in fact started with a single population, extended to several populations. By 1985, the resultant germplasm has been systematically introduced and further adapted to conditions in Africa since 1996, and more recently in Asia. This is one of the major reasons why CIMMYT now has an extremely strong germplasm base for an array of stresses. Coupled with this is an extensive network for managed stress screening of important abiotic as well as biotic stresses. Here I present to you only Sub-Saharan Africa and Asia. A similar network also exists in Latin America, but what is really impressive are these managed stress screening locations for drought, drought plus heat, heat alone, low nitrogen plus drought, as well as TRIGA in partnership with IITA, our sister center in West Africa. Similarly, in South and Southeast Asia, again a network of sites available for drought, waterlogging, heat, as well as cold stress tolerance phenotyping. And this is something which happened in the last 3 years. In fact, the largest tropical nitrogen use efficiency screening network in Africa. We started with a very small level in Ghana in 1986, but today over 100,000 low nitrogen plots are available for screening of germplasm in as many as 15 locations in 6 Eastern and Southern African countries, including the confined field trials for testing the transgenics that are developed through the IMAS project. Bish Das has done remarkable work in establishing this network. And this is important for us since nitrogen deficit accounts for almost 15% of the yield gap in the tropics, especially in the warm lowlands of Sub-Saharan Africa. This network, these partnerships, this has ensured that there is a continuous pipeline of stress-resilient germplasm that have been introduced in the tropics and adopted in the tropics. The flow is very, very important, and that is practiced across the Global Maize program. Managed stress screening for relevant abiotic and biotic stresses in the target population of environments is an integral part of our breeding process. And the best products that come out of this across different projects are tested through integrated regional trials managed by CIMMYT for identifying the best bet products. And from those regional trials, we again pick up the best products and undertake extensive regional on-farm varietal trials. This is called ROFVTs, and against the best commercial checks in those environments. And through this rigorous process, what we get is nominated as entries by both public and private sector partners through the national performance trials, varietal releases, seed scale-up, and then adoption. How does that results look like, especially for drought tolerance? This is a project called Drought Tolerant Maize for Africa, with extensive work in 13 sub-Saharan African countries. And the drought tolerant products that come out of CIMMYT, and when they are tested under high yield trials, that means more than 3 tons per hectare locations, these are regional on-farm varietal trials. And these are low yielding trials under low input conditions. You can see here the CIMMYT hybrids, they show extremely good response even under drought stress. At least a 20 to 40% improvement over the best commercial checks like Seedco's or Pristine or Panar or Pioneer hybrids in Africa. The same holds good. That means these are not only performing under random drought stress very effectively, but even under high yielding trials, the percent yield level against Seedco 5 and 3 could go as much as 25%. So the operation is not only successful, but the patient is also living and thriving under optimal conditions and not just under drought tolerant conditions. And that's the success of the products from CIMMYT in Africa. The result is today we have 140 new drought tolerant varieties that are released in Africa in 13 countries, including 81 hybrids and 59 improved varieties since 2007 through a single project. One of the most phenomenal achievements one can ever talk about in terms of varietal deployment. And today 30,000 tons of drought tolerant maize varieties are produced by partners, and this is done by 51 companies which are on our list of DT variety producers, of which 44 are producing exclusively CIMMYT-derived drought tolerant germplasm, and the rest producing combination products. That means CIMMYT lines plus their own lines in the varieties. In total, an estimated 1.23 million hectares are planted right now in Africa with these new DT varieties, benefiting about 3 million households. So conventional breeders have been quite successful, even without extensive application of molecular markers. The strength of the germplasm, the strength of phenotyping is extremely critical in terms of the success. But what can markers do is what I will come to later. But this is something the nitrogen use efficient donors have come from the IMAS-Association mapping panel for nitrogen use efficiency, improved maize for African soils. We have an array of very highly effective nitrogen use efficient donors coming out of this project. This is one of the important products of association mapping. Just don't think that GWAS can only lead to marker discovery. GWAS, if it is applied properly and with proper phenotyping, can also lead to identification of excellent donors for important traits. That's one of the important things that is to be stressed when we talk about genome-wide association studies. Besides that, we have the largest genetic or genomic resources and a database with high-density genotypic information in tropical maize that extends to more than 30,000 samples of CIMMYT genotyped with GBS, genotyping by sequencing. And these are all the list of a range of populations or panels or elite lines that have been sequenced, that have been genotyped using GBS over the last 2-3 years, and this list is growing very rapidly. The second aspect is how to manage that information, and this is something again we are struggling to cope with, and both Andres is here and John Marcel is there, and these are our resource person in terms of dealing with this tsunami of genotypic data in maize, in terms of billions of data points generated on some of the most important tropical and subtropical populations. What I will now focus on is about a robust trait pipeline. Now, since the last 3-4 years, we have been intensively focusing on a number of important stresses that affect tropical maize productivity. These are now in 3 different phases. One is the discovery phase. You have waterlogging, heat stress tolerance, banded leaf and sheath blight, phosphorus use efficiency, tar spot, and striga. We now have under validation phase some very important traits. Drought, nitrogen use efficiency, maize lethal necrosis, constant complex, gray leaf spot, and Tarsicum leaf blight. We already have under the deployment phase 3 very important traits, that is provitamin A enrichment, maize streak virus resistance, and a very important QTL for haploid induction rate. I will talk about each of this. Let's first start with the deployment aspect. This is one of the most successful examples of allele mining leading to marker discovery, validation in several biparental populations and breeding germplasm, and resulting in products that have been deployed for the farmers. I haven't seen many such examples in the literature. This is come through a very, very important study undertaken through Harvest Plus, where 2 major genes, lycopene epsilon cyclase and carotenoid beta hydroxylase, natural allelic variation for these 2 genes have been identified. And this is important because while this pathway needs to be proceeding well and beta carotene needs to be accumulated in the kernel, this pathway of lutein accumulation has to be brought down and the resources diverted into this branch. So that the selection for these 2 genes or favorable alleles for these 2 genes is very critical for higher accumulation of provitamin A. And if we don't apply marker-assisted selection, the average Pro-A levels through conventional breeding could go not more than up to 8 ppm. White maize has 0 ppm of beta-carotene. In yellow commercial varieties which you see in the market, it can go anywhere between 1 to 3 ppm. So through simple phenotypic selection, you can go up to 8 ppm, but if you use MAS for these genes, you can take the average provitamin A levels up to 35 ppm. So we don't require golden maize here. You can simply use marker-assisted selection for developing high provitamin A enriched maize. And this is being done, but what Raman Babu has done systematically is to ascertain how these polymorphisms work in a range of germplasm, and the best performing assays has been developed by him. And now we very routinely use markers for only carotenoid beta hydroxylase in our pedigree or back crosses in the CIMMYT Harvest Plus program. We now have next generation provitamin A enriched lines. Till 2012, the provitamin A enrichment depended upon only 2 lines from 2 elite donor lines from IITA. But today, thanks to this work, allele mining of favorable alleles for CRT RB1 was done in 64 tropical, subtropical lines, and a large number of lines are now available with us, and widening the choice of using these lines in provitamin A enriched program. And tremendous work done by Natalia, Babu, and the Harvest Plus maize team. Another very successful example for how fine mapping for an important QTL can lead to production markers that could be used in breeding is maize streak virus. This is a very important disease indigenous to Africa. At least 20 countries in Africa have reported devastating epidemics of MSV in the past. And these epidemics are more common to occur along with drought, which is a very frequent phenomena in sub-Saharan Africa. And therefore, without MSV resistance, you cannot have a successful product, especially in Southern Africa. Phenotyping is very cumbersome, and that's the reason we went ahead and Sudha Nair and CIMMYT-DTMA team have done excellent work. It is in fact the first fine mapping work done on disease resistance leading to production markers now under deployment phase. Best haplotypes have been identified, and DH lines have been screened against this, and one can see here the prediction, the accuracy of using the resistant haplotype is almost 0.94. It's a tremendous success even in a complex crop like maize. And now we have pilot deployment of both provitamin A enrichment and MSV resistance in some of the best germplasm in sub-Saharan Africa. Okay, the markers in maize, we all know now, can be even more powerful when they are combined with double haploids. And this double haploid work has been severely constrained in tropical regions because of lack of proper haploid inducers. And this bottleneck has been overcome more recently in collaboration with University of Hohenheim. Now we have tropical haploid inducers of CIMMYT, which are far, far better than the temperate inducers, which hardly lead to any pollen, which are severely vulnerable to diseases or insects in tropical conditions. We have improved CIMMYT lines with the haploid induction property as much as 8 to 10%. And these inducer lines we are already started distributing to institutions worldwide for research use or for commercial use. And the recipients come from diverse countries across Africa, Asia, and Latin America. More importantly, we started double haploid service on a cost recovery basis to our NARS and small and medium enterprise seed companies in Africa, Asia, and Latin America by establishing very strong DH facilities. This is a state-of-the-art maize double haploid facility established in Kibako, Kenya in 2013 September, which can produce as much as 120 to 150,000 double haploid lines each year and suffice to meet the major requirements of the programs there. And what is very exciting is the discovery of a major QTL for haploid induction by 3 different groups on chromosome 1 way back in 1997, and then 2008, and more recently Vanessa Preg, Hohenheim student working in CIMMYT, identified this in the materials that came from Hohenheim. This QTL region was, again, in very recent 2013, was fine mapped to a region, an interval of 11.6 centimorgan. Sudha, who is based here in ICRISAT campus, CIMMYT's employee, along with Vijay Chaitam, our DH specialist, were successful in identifying 2 markers that flank this QTL at a 3 centimorgan interval, along with 2 internal markers. And these are sufficient enough for using this QHAR1 in our marker-assisted selection schemes. And this is done using the progeny test mapping strategy, a very, very cumbersome way of understanding how haploid induction rate should be maintained, but at the same time, validation of this using significance testing of the plus or plus minus or minus classes within each large F3 families. And through this exercise, the QHAR1 interval has been delimited, and then these markers are also validated in different biparental populations, which we are currently using for 2nd generation inducer development in CIMMYT genetic backgrounds. Remarkably, you will see here that if the number of families without QHAR1 and with almost 0 to 3% negligible levels of haploid induction rate are 93.75. That means whereas you can see QHAR alone may not be guaranteeing high haploid induction rate, but if you have to have high haploid induction rate that have to cross 8%, you need to have QHAR. Very beautiful case of how fine mapping can lead to an excellent way of understanding how it relates to the phenotype, even a complex phenotype like haploid induction rate. So this is now a very validated marker, and it's already being deployed in diverse continents for including sub-Saharan Africa, Asia, and Latin America for the generating 2nd generation haploid inducers. Coming to more complex traits, so those are our 2 simple cases: provitamin A, maize streak virus, and haploid induction rate. Coming to drought, what's the situation in drought? So we started off in the last 3-4 years by extensively evaluating a panel called the Drought Tolerant Maize for Africa DTMA-Association mapping panel, comprising roughly around 300 lines. These lines have all been genotyped initially with 55K SNP chip and later with GBS. And this panel comprises lines from Colombia, Ethiopia, the Highland program, the physiology, some of the very good lines coming from John Marshall way back when he was a student. He developed some excellent lines for drought tolerance. They are all included in this. Subtropical, tropical, IITS, drought and striga tolerant lines as well as CIMMYT Zimbabwe lines. And this has been extensively phenotyped. One of the best panels for drought tolerance screened for drought, soil acidity, heat, drought plus heat, waterlogging, and several of the major diseases of maize existing in Africa, Asia, and Latin America. The population structure in DTMA-AM panel looks like this, with a clear separation of Laposta secchia, the DTP, the CIMMYT-Kali lines, and here are the subtropical lines. But what is remarkable here to note is that unlike the temperate maize where the LD decay distance is much, much larger, the LD decay in the tropical germplasm could be happening very highly. There is almost just 2.13 kb here in the DTMA-AM panel. So this is a very, very important factor to note. The tropical, subtropical germplasm show a much stronger LD decay as compared to the temperate germplasm. But what we got as initial leads from extensive evaluation of this AM panel have been recently validated through several, it's not just a discovery, 8 genomic regions have been identified to have significant positive effects when CIMMYT's drought donors were analyzed. These have all been characterized for their haplotypes, and Babu and the DTMA team validated this across different populations and panels, including an ABQTL Laposta secchia population, DTP population, F64 derived double haploids, DTMA Mexico MARS biparental populations, as well as the WEMA-DT population, as many as 18 biparental populations. This is the extent of effort of validating what comes out of the GWAS leads. And through this exercise, we have identified some very major regions, especially on chromosome 1, 5, and 10, validated in as many as 4 of the 5 resources. And Babu is now exploring opportunities to use this as potential targets for introgressions in some of our susceptible but very good lines. Joseph BNA of CIMMYT published a paper recently on genetic distance among the double haploid maize lines and their test class performance under drought stress. This is a very important resource, genomic resource for us. Several BC1, F1 source populations have been derived by crossing 4 different drought-tolerant donor lines, including Laposta secchia and a temperate source, with 4 recurrent parents. 806 double haploid lines have been generated in this, and this is an ideal population for backcross plus double haploids plus genotyping by sequencing. That's a very, very ideal combination for dissection of some of the complex traits like drought tolerance. And Babu made use of these 806 DH lines derived through these specific sources and dissected drought tolerance in these backcross derived DH lines. And identified some very good genomic regions from the 4 DT donors. What's remarkable is the way in which you can characterize how these CML395 derived double haploid test classes can differ from 488 derived one and 312 SRs or 444 derived test classes. Distinct haplotypic, distinct genomic regions identified in each of these. And native variation therefore exists with significant positive effects for some of the major genes that contribute to drought tolerance in CIMMYT's drought donors. And Babu has developed the 30 breeder-ready assays, the Caspar SNPs, to facilitate introgressions in this. Through WEMA, a water efficient maize for Africa project coupled with DTMA, today CIMMYT has the world's largest resources for undertaking GWAS and genomic selection in tropical maize, as many as 28 biparental populations, with 5,000 lines in total, each genotype initially with 160 to 350 SNPs. And to link the data from WEMA and DTMA, approximately 100 lines per populations also genotyped by GBS. And the GBS and phenotypic data is now being used for finding and validating genomic regions for drought tolerance as well as for implementing pilot genomic selection. This we have done through what we call as genetic gain studies by systematically doing marker-assisted recurrent selection for cycle 0 to 1 to 2 and 3, crossing it with a single cross tester, taking these single cross tester hybrids and comparing them widely against the best commercial checks. Joseph BNA has done a remarkable job on this. And you can see here what is the genetic gain that we can obtain using marker-assisted recurrent selection. Dr. Gupta in his opening remarks said that most people use only marker-assisted backcrossing, and not many institutions are doing marker-assisted recurrent selection or genomic selection. That's why, Dr. Gupta, this is the slide for you to show the hybrids developed from markers-only selection till cycle 3 have produced on an average 2.4 to as much as 21.7% higher yield than the base population under drought as well as optimum environments. And this is just a study that has been recently completed 2 weeks back. And if you compare the hybrids developed from Mars versus pedigree-based selection, you can see as much as 3.9 to 16% increase under optimum conditions, and under drought conditions 1.8 to as much as 50%. So friends, the message here is that marker-assisted recurrent selection can indeed work for improving drought stress tolerance, grain yield under optimal as well as under drought stress environments. Similarly, we have another excellent panel called IMAS, Improved Maize for African Soils Association Mapping Panel. Here, some germplasm has also contributed by ARC in South Africa. And ARC has a distinct breeding history for temperate germplasm. So you have a distinct separation of Mexico-based lowland tropical germplasm. You have subtropical germplasm in the middle, and the ARC temperate germplasm here. And this panel has been genotyped again by 55K plus GBS, phenotyped in 6 low nitrogen plus 13 low nitrogen locations in, till 2013, plus a variety of other traits. What does this show? Babu has again some very interesting results, very interesting leads on what genomic regions can potentially contribute performance of maize genotypes under nitrogen stressed environments. He is presently validating them in several biparental populations, but I just want to show you here how Babu is trying to use these different structure groups in terms of benefiting from nitrogen use efficiency. He identified 5 genomic regions in CIMMYT germplasm for grain yield under low nitrogen conditions, and the favorable alleles for this, if you look at those alleles in the ARC and CARI germplasm, Kenya Agriculture Research Institute germplasm, they carry completely unfavorable alleles for those 5 genomic regions. That means by understanding the structure groups within this IMAS panel, one can take a targeted introgression of definite favorable alleles from CIMMYT germplasm for improving even temperate sources like those present in South Africa. And very good introgression targets that can lead to as much as 0.19 tons per hectare to as much as 0.38 tons per hectare. Each of these SNPs can potentially contribute. Yes, so we also have a strong pipeline of water stress resilient Asia adapted germplasm, which is not only ready for deployment in rain-fed ecologies, but providing an ideal platform for intensive genetic analysis of drought, heat, and water logging. A range of heat-tolerant donor lines have been identified, and this showed very clearly that drought tolerance in tropical germplasm doesn't always result in heat tolerance, and lines which are tolerant to either of these two stresses, when it comes to drought plus heat, can completely collapse. So this is a very, very important message for tropical breeders to look at it. I won't go into the details of maize lethal necrosis. This is a very, very important disease that is coming up in Africa, rapidly spreading from Kenya, Tanzania, and Uganda, and CIMMYT has identified DT plus MLN resistant maize germplasm, probed the genetics of resistance to MLN, and identified 3 major QTLs that are exactly parallel to chromosome 6, 3, and 10. 1 minute on my final messages. We need to further enhance genetic gains in stress-prone environments. As Dr. Gupta has pointed out in his remarks, high-throughput field phenotyping is really the need of the hour. Not just in the phenomics facilities, but actually field-based phenotyping that can be used by breeders. And that's what CIMMYT colleagues are presently doing in terms of understanding how to take empirical decisions on imposing drought stress, monitoring and factoring weather data, as well as semi-automated non-destructive measurement of root rates. And more recently, a customized unmanned aerial vehicle is now being used in Zimbabwe with the multispectral and thermal image cameras, which can take instant images of our entire maize fields that are subjected to drought stress or low nitrogen tolerance stress. And this can be an excellent tool for heat stress phenotyping. But what's more important is we need to understand how to manage this data. This is a tremendous amount of data. In conclusion, genomics assisted breeding can be very impactful, effectively implemented for developing and delivering stress resilient germplasm. When we effectively integrate well-targeted managed stress environments with well-run multi-environmental trials, precision phenotyping, double haploid tools, as well as decision support systems. Use not only proven sources of stress tolerance, but widen the search for these stress tolerant sources through projects like Seeds of Discovery, enhance the repeatability of field-based phenotyping, identify and promote relevant agronomic practices that exploit improved genetics, increase access and affordability of stress-resilient products. That's the ultimate proof of all our efforts. Otherwise, no amount of basic work on genomics can really alleviate the suffering of the resourceful farmers. Through vibrant seed systems and policies, and promote public as well as public-private partnerships. There is a saying in Africa, Richard Hartgett says that every morning in Africa, a gazelle wakes up. It knows it must outrun the fastest lion or it will be killed. Every morning in Africa, a lion wakes up. It knows it must outrun the slowest gazelle or it will starve. It doesn't matter whether you are a lion or a gazelle. As the sun comes up, you would better be running. Don't run to the gala dinner, but thanks a lot to the CIMMYT colleagues, partners, and funding agencies which support the work of CIMMYT.